So let's start with the next part of microbiology, and that is bacteriology, something which scares the students a lot, not only because of its bulk, but also because it's very, very volatile. So we'll try our level best so that we can simplify this with the help of some mnemonics and some clinical integration. Before I begin with bacteriology, the first rule that I want to tell all of you, there are two chapters, cocci and bacilli. And by definition, all the cocci are going to be gram-positive and the bacilli are going to be gram-negative. But there's nothing 100% in medicine, right? So if I'm saying that all the cocci are gram-positive, I agree. But then there is a mnemonic that is called as venom. We know venom matlab there's going to be something very negative, something very deadly. So venom means that when we are talking about Neisseria, when we are talking about Moraxella and Veillonella, these are the three bacteria which are cocci but are exceptions. They are actually gram-negative cocci because venom is something very, very negative, right? So gram-negative, rest all of them are gram-positive. So that is how we'll proceed. First we'll do all the gram-positive cocci, then we'll do these gram-negative cocci. So starting with the usual classification of the GPC, the gram-positive cocci. Guys, we divide it into two families. One is referred to as the Micrococcaceae family, and the other is referred to as Streptococcaceae family. These are the families that I'm telling you, and don't worry, in the entire microbiology, I know you all are very scared because many of the places you've read so many families, you don't have to learn all. You have to learn certain families, and that I'll tell you as and when, like this is one of them. So under Micrococcaceae family, you don't get any points for guessing that one of the bacteria is going to be Micrococcus, but the other cousin brother of it is going to be Staphylococcus. The second one we have is Streptococcaceae. Of course, this is going to have all the streptococci that you ever have heard of. Streptococcus pyogenes, Streptococcus agalacti. In fact, there is something known as Streptococcus pneumoniae, which many people know as pneumococcus. Of course, that will also come under it. And then there is an organism called Enterococcus, which many people are saying can be put into this category. Some say it is a separate family. But abhi ke liye, Micrococcaceae has micro and staph. Streptococcaceae has all strepto things and Enterococcus, and that is what I'll start my chapter with. I'm starting with the Micrococcaceae family, and the first is the difference between staph and Micrococcus. So let's get going. I know it's difficult, but we'll try. Guys, do you know what is the meaning of the word staphyle? Staphyle ka matlab hota hai bunch of grapes. So which is the bacteria that looks like a bunch of grapes, and that is going to be Staphylococcus. What does Micrococcus look like? Micro chota tetrads. They are going to be bunch of four. Only a bunch of four that is going to be tetrad. So repeating, Staphylococcus is staphyle grapes and Micrococcus tetrad. Then you're going to see that what is common between them. Common point here that both of them come out to be catalase positive. That is their commonality. That is why they are under one family. They are catalase positive. But then we did the difference in terms of oxidase. Dekho, Micrococcus is oxidase positive, Staphylococcus is not. And how did we analyze it? You will ultimately realize that Micrococcus is gonna have emphasis on all these O's. Micrococcus में micro na, so micro में छोटा सा O, zero, anda O, छोटा, that is how you will remember. So oxidase, all the O wala things are gonna come up over here. It is oxidase positive. Similarly, in glucose utilization, if they ask you which of them is having an oxidative pattern and a fermentative pattern, you will say common sense, Micrococcus has an oxidative pattern, and Staphylo में वो F wala sound आता है ना, Staphylo has F wala sound, so that is gonna have a fermentative pattern. ठीक है? So now you've learned it, guys, that whether you are asked about glucose utilization being oxidative or you're asked about what is oxidase positive, Micrococcus will always give emphasis to O, and Staphylococcus is always going to give emphasis to F. So it is fermentative, it is not oxidative. Next, what is its response to bacitracin? So now tell me which of them is sensitive and which of them is resistant. Resistance matlab tough. Resistant means it's tough. Now, obviously, I'm changing the spelling. When I say tough, matlab F wala sound is coming. Staphylococcus is tough to bacitracin, matlab resistant, and Micrococcus is gonna be sensitive. I hope everyone's understood this, learnt it, mugged it up. इसमें there is no concept involved, but there is definitely a lot of mugging up. So Micrococcus में सब कुछ OO आएगा, oxidative, oxidase positive, and sensitive. Staphylococcus has fermentative pattern, no oxidizability, and it's gonna be very, very tough, matlab resistant. Now please note something important which could come up in the INI CET exam. What is the medium that we use for the glucose utilization? We use the Hugh and Leifson's medium. there is something known as Hugh-Lifson medium, and for Hugh-Lifson medium we have bromothymol blue as its indicator. Bromothymol blue is gonna be the indicator that is involved in the Hugh-Lifson's medium. That finishes off the difference. So we are starting now individually, guys. The main organism here to study is Staphylococcus, and I've already told you Staphylococcus aureus is what I'm starting with. So kya kya pata hai? Tell me one thing. Staphylococcus had its emphasis on F. So is it going to have anything oxidase? Oxidase? No. Ma'am, it is oxidase negative, and catalase, so the entire family is positive. So these 2 points I already knew. After that, you did not tell me anything about motility, anything about sporing. Koi flagella nahi hai, spore nahi hai, no motility, no sporing. And we've already told you that staphyle word means bunch of grapes. So yes, this is exactly how it's going to look like, which I'll show you in a while. But why is it called Staphylococcus aureus? Guys, staphyle word you've understood, bunch of grapes. Coccus because ye hai gram-positive coccus. Aureus, go back to your chemistry days. Au is the chemical formula of what? Gold. Remember, this produces a golden pigment. This is going to produce a golden pigment, which is very, very important. ठीक है, Staphylococcus हो गया. Should we start with the virulence factors? So every bacteria will be divided, virulence factors, then clinical features, diagnosis and treatment. Starting with the virulence factor, something that will be present in the cell wall, then cell surface, then toxins and enzymes. So cell wall में क्या होता है? I think everyone's cell wall, go back to your general microbiology, you know that the gram-positive cell wall has a lot of peptidoglycan. Gram-positive organism है ना? So a lot of peptidoglycan will be there. Also, do you remember all the gram-positive organisms have teichoic acid? This also I've taught you. So teichoic acid was for what? What's your mnemonic? Teichoic acid is for ticna. Ticna means attachment. So whenever we are going to have gram-positive organisms trying to attach, teichoic acid is for ticna, right? Attachment. These are cell wall related. What is cell surface related? Cell surface related, we have protein A and clumping factor. Both of these I'm going to talk about in a while. But abhi ke liye my focus is going to be on protein A, because please note when you write A over here, it has all the A wale works that it's going to do. It's anti-complementary. Means, is it going to allow your complement system to work? No. It is anti-phagocytic. Means, will it allow your phagocytosis to work? No. So kind of it's good for the bacteria, na? If you don't do phagocytosis, you can't kill it. If you don't do complement system, you can't kill it. So bacteria is getting its protection with itself. Moving on, coming to the next one, tell me something which is known as coagglutination. Now, what is coagglutination? This is exactly what I'm going to explain to you. But pehle please learn, protein A is responsible for anti-complementary, anti-phagocytic and coagglutination. So what is coagglutination? Sabse pehle, this is a test where I want to detect antigen. You'll say, hang on ma'am, Staphylococcus is anyway an antigen. Are you trying to detect another antigen? Yes. I am using Staphylococcus to detect something else. So what will I do? I will take the Cowen-1 strain of Staphylococcus. Listen to these two sentences very carefully. You will take what strain? I will take the Cowen-1 strain. That's a question. What strain is used for coagglutination? Cowen-1 strain. And every strain of Staph aureus, it has protein A. So I'll assume, ठीक है, protein A है उसके अंदर. You know what protein A does? Protein A binds to IgG. Can you see these antibodies? Which part of IgG has protein A gotten bound to? It is bound to the Fc segment of IgG, which means these segments are free. FAB, antigen binding wala segments are free. So if I put any other antigen like a salmonella or any other antigen, that antigen will easily be caught and ek bada sa clump ban jayega. There is going to be a big clump. So please remember, whenever you're talking about Staph aureus, which strain? Cowen-1 strain. Staph aureus has protein A. Protein A will bind to IgG. IgG ka kaun sa portion? FC portion. So the FAB portion is free. Now I can attach any other bacteria or any other antigen onto it. This means there's going to be a big clump, ek staph wala clump, some other bacteria clump. Big clump will form, and that is now going to be referred to as coagglutination, which is very important. I hope everyone's okay with this. You understood? Protein A, anti-complementary, anti-phagocytic, coagglutination. Done. Clumping factor I'll come to in 5 minutes. Okay. Now let's move forward. The next is that what are the toxins that are being used? And I think this is something which everyone has heard of. The first set of toxins that we have are hemolysins. They will break the blood. So we have alpha, beta, gamma, delta. Alpha, beta, gamma and delta. 4 hemolysins. You will say, what blood is it breaking? So I will now tell you that this is going to lyse sheep, and human blood. Okay, then even gamma is going to lyse sheep and human, delta is also going to lyse sheep and human blood, but your beta sphingomyelinase wala jo toxin hai, that is going to lyse the sheep RBCs only. Why is that so? When everyone else is ready to lyse sheep and human RBCs, why is this beta a little different? That is why beta has been given another name, na, sphingomyelinase. It breaks sphingomyelin because sheep RBCs have high levels of sphingomyelin. Sheep RBCs have high levels of sphingomyelin, and that is exactly what it intends to break. That is why the beta hemolysin is known as sphingomyelinase. यहाँ तक समझ आ गया? All of them are breaking all the RBCs. Beta sphingomyelinase, S wala S, so it will lyse only the sheep RBCs. What else? This alpha 1 has a little bit of a temperature problem, means it is going to get inactivated at 70 degree and activate again at 100 degree. So alpha wala says that I am going to go at above 100 degree and then get activated. So somewhere it is going to get activated. How did I learn it? Alpha wala or A is going to get activated at 100 degree. Okay, so alpha activated at 100 degree, not below that. At 70 degree it will get inactivated. Okay, what is beta sphingomyelin is showing you? It shows you a very, very important thing known as hot cold phenomena. Hot cold, you really don't have to know the definition, but I'm एक बार सुन लो, ठीक है? Hot means it's going to work at hot temperature. Cold means it's going to work at cold temperature. What comes first? Hot comes first. Means at 37 degrees Celsius it will start working, and cold means at 4 degrees Celsius it will complete its work. ठीक है? तो hot पहले आया, cold बाद में आया. At hot 37 degree it will start its work. At cold 4 degree it is going to complete its work. That is what is the hot cold phenomena. So out of this slide, what are your PYQs? They ask you which is the one which is known as sphingomyelinase. It is beta 1, S for S. It lyses which RBCs? Sheep RBCs. Which are the ones which show you hot cold phenomenon? Again, beta sphingomyelinase. So beta hi sabse, beta is most important over here, right? This was only one, the hemolysins. Abhi the story is not over. What are the other toxins that you have? The second one is the PV toxin, Pantin-Valentine toxin, and this is usually seen with all the methicillin resistant Staph aureus. The MRSA wale strain, which I will be telling you in a while, the MRSA strains show you PV toxin. Now I'll tell you another mnemonic. PV toxin is known as synergohymenotropic toxin. What is synergo? Synergo means someone is working in synergy. दो लोग एक साथ, if two people work together, that is synergy. So remember, PV toxin is synergohymenotropic. And out of these alpha, beta, gamma, delta that I just told you, the gamma one is going to be synergohymenotropic. कैसे याद करें? Because they will say which of the following toxins are synergohymenotropic. PYQ. चलो PYQ करो. So you will have PV तो याद रहेगा, but then you will get confused. What is was it alpha, beta, gamma or delta? So the way you write PV, but PV के ही continuation में you guys write gamma. They have to work together ना, synergy maintain करना है. So synergy maintain करने के लिए you will write a gamma over here. So PV and gamma hemolysin are said to be synergohymenotropic. Coming to the next toxin, the name is only self-explanatory. Epidermolytic. It is going to break the epidermis. Skin ka problem hai. Skin is gonna get exfoliated. So of course they ask you that which is the staphylococcal toxin which is gonna cause staphylococcal scalded skin syndrome. Of course, staphylococcal scalded skin syndrome is caused by staphylococcus, and skin ka problem hai, so it must be the epidermolytic or exfoliative toxin. When the same thing occurs in children, what do you call it? That is known as the Ritter's disease. The Ritter's disease, right? And sometimes the staphylococcus can also cause another skin problem known as toxic epidermal necrolysis. Toxic epidermal necrolysis, which I hope you've read, it happens because of a lot of drugs also. That can happen because of staph as well. So जो भी skin से related problem पूछेंगे ना, SSS, Ritter's, Sten, that is going to be by which toxin? Epidermolytic toxin. Now one more question that you get in dermatology. In staphylococcal scalded skin syndrome, what is the site of action for the mechanism? Which part of the skin does staphylococcal go and attack? This is going to go and attack the stratum granulosum. And which antigen in the stratum granulosum? It is going to be desmoglein 1. The first one is going to be attacked. This is important mainly for INICT and dermatology में बहुत important है. That is in SSSS, what is happening? There is going to be attack of which part of the skin? Stratum granulosum, desmoglein 1 is attacked. Okay, now the toxins are still not over. PV हो गया, epidermolytic हो गया. Now let's come to entero. मतलब entero, ma'am, food poisoning वाला toxin. Exactly. Which are the food poisoning वाला? A to I, but no F. Remember, F को ना females के लिए रखो. For the time being, keep F for females. Something in females I will tell you. So A to I, but not F. Then R to T and V. If you can learn these alphabets, very good. If you can't, at least learn this much, all except F. बाकी सब food poisoning कराएंगे, not F. Okay. So now what am I saying? A to I, out of which enterotoxin A, of course, comes first, is the most common. And these are heat stable toxin. Heat stable means that even if you're heating food, for example, if you are, heating milk, or you're heating some meat or some poultry, even after heating, they will survive. That is why even after having hot food, someone can have this food poisoning. So what did I say? The person is going to have food poisoning. Now you'll ask me, ma'am, which kind of food poisoning is it? Is it the vomiting wala food poisoning or the diarrhea wala food poisoning? So usually it's vomiting, because this toxin has a vagomimetic action. It tends to stimulate the vagal stimulation happens. So there is going to be emesis, there is going to be vomiting. So what all did you learn? All the toxins, entero word se the food poisoning will come to your mind. Even if the food is heated up, it will happen. And that is going to be vagus stimulation, so patient will vomit out. All the toxins are going to be responsible, but not F. And out of them, A is the most important. What is the incubation period of this food poisoning? The incubation period is between 1 to 6 hours. So please remember, you get a question in the paper that there is a food poisoning or vomitings which are happening within 1 to 6 hours of consumption of some food. Either you're going to think of Staph aureus or you're going to think of Bacillus cereus. दोनों का incubation period for vomiting is 1 to 6 hours, okay? But remember, for Staphylococcus aureus, you are going to have consumption of milk and poultry, or poultry you can learn as meat. You're going to have milk and poultry consumption. I'll give you an example. Previous year question was that there were 20 people who went for a birthday party, okay? Out of them, 19 people, because they consumed creamy pastries. What is creamy pastries? Creamy pastries means milk, hai na? So they said that 19 people consumed creamy pastries after that birthday party, and as soon as they got back home, within 6 hours, they started having vomitings. What is the suspected organism? So jaldi wala food poisoning, and because of milk, meat, poultry, that is Staph aureus. Whereas Bacillus cereus, C for C, that is, if they give you a history of Chinese fried rice, then they are referring to Bacillus cereus. That is going to be your hint in the exam. So milk, meat ke liye Staph, and C for C, Chinese fried rice, cereus. Both of them have an incubation period of 1 to 6 hours. Okay, so what did I tell you? All toxins, but not F. So what is that F wala enterotoxin? Something in females. Please note that is gonna cause, or that is also known as the toxic shock syndrome toxin. Toxic shock syndrome toxin, that is why we call it TSST, and that is enterotoxin F. Why did I say F for females? Because this is associated with long-term, this is forgotten what? This is associated with long-term tampon usage. So tampons are obviously used by females during menstruation, so there will always be a history of tampon usage. It can happen in males also. There can be, non-menstrual causes also of toxic shock syndrome. But 99% of the cases, this is the history that you're going to get. That is why I've made you learn it like this. That enterotoxin F जो है वो कुछ female से related होगा, tampon history होगा, menstrual history होगा. Okay, now why is it so deadly? Not only am I going to say F for female, I am also going to say F for fatal, because this is a very deadly disease. Why is toxic shock syndrome so deadly? Because this antigen is not a regular antigen. I hope you remember from your immunology chapter, this is going to be a super antigen. So this is definitely something which is very, very important for you to know. Okay, guys, I want to take you a little bit into medicine. Do you know what is toxic shock syndrome? Quickly, we'll revise our criteria from medicine. What is the criteria? Kitna fever hona chahiye? Fever should be above 102 degree Fahrenheit, right? Okay, along with that, there should be hypotension. Hypotension means systolic blood pressure is less than 90 mm mercury. So remember, fever above 102, BP less than 90, and a rash that is present. This is like definitive. Plus any 3 of the following organs. तो क्या क्या है? GIT में क्या problem होगा? GIT is always our favorite, nausea, vomiting. Kidney, if you want to assess, measure the serum creatinine. If the serum creatinine is going to be more than 2 times the normal, problem hai. Liver में best is measure SGOT, SGPT. If they are more than 2 times normal, problem hai. Central nervous system में best is if the person is disoriented, confused. Then for muscle, if the CPK levels are more than 2 times normal, again that is going to be a problem. If the mucous membranes are coming out to be very, very red, that is a problem. And coagulopathy means the platelet count is less than 1 lakh. I hope you remember from your path and medicine, normal platelet count starts from 1.5 to 4.5 lakhs. मतलब 1.5 lakh तो starting है. Here the platelet count has gone below 1 lakh also. So that is going to be a coagulopathy. If any 3 of the following organs are affected, and this was a very easy way of learning, जो भी organ होगा उसका whatever problem is there has to be 2 times normal. Okay, twice the normal range, and that is going to be a problem, right? So that is toxic shock syndrome. I hope everyone's clear with the toxins, because I want to take you all the way back to the virulence factors, guys. Under that, I told you once you're done with the toxins, kya kara? You did the hemolysins, alpha, beta, gamma, delta. Do you want to make a quick list? Under the toxins, you had the hemolysins, alpha, beta, gamma, delta. Then you had the pantin-valentine toxin. Then you had the one that was affecting the skin, so epidermolytic toxin, staphylococcal scalded skin syndrome. Then you had the food poisoning wala, that is enterotoxin, all except F, because wo jo fatal disease tha in females, that was toxic shock syndrome toxin. That is a list of your toxins. Means now you're ready to take up the list of your enzymes, which is super simple because I have a mnemonic for you. The way you write staphylococcus, you're going to take up some alphabets out of staphylococcus and get your answer. What are the enzymes that it makes? With T, there is going to be thermonuclease, and nuclease ki baat aayi, so I can also write DNase. T for thermonuclease and DNase. PH is going to be for phosphatase. PH will be for phosphatase. Repeat, T for thermonuclease, DNase, PH for phosphatase. Then there are 2 C's, and there are no points for guessing that these are the most important, that is catalase and coagulase. Catalase and coagulase. So later on when I tell you the diagnosis, I will show you they are catalase positive and coagulase positive. Okay, enzymes are done. Let's move on to the clinical features. That also has a mnemonic called soft pains. What is soft pains? So S is going to stand for all the skin and soft tissue infections. Now guys, there are so many skin, I mean that's such a vague term to use, na? Skin and soft tissue infections. That means you could have abscesses, you could have furuncles, you could have cellulitis, so many things possible. I'll give you just one example which is really asked in the exam, and that is the example of botryomycosis. Sounds like what? Mycosis sounds like a fungal infection, and with this kind of a picture, botryo, what do you mean by the word botryo? Botryo again means bunch of grapes. Yes, this is looking like a bunch of grapes. So with this kind of a picture, I'll start thinking of Madura foot. I will start thinking of fungal infections and all of that, hai na? But please remember that this botryomycosis is something which is differentiated clinically. Confusion hoga. You will think of Madura foot and all of that, but this botryomycosis is something that is gonna be differentiated by Staph aureus. This is caused by Staph aureus, so it is very, very different from the others that have been shown. Now let's move forward. S for soft and skin infections. O is going to be in orthopedics. You've read osteomyelitis. The most common cause of osteomyelitis is Staph aureus. Okay, then F is going to be what you just now studied. There is going to be food poisoning, and you know the incubation period of that food poisoning, it's going to be less than 6 hours. Short-term food poisoning. Okay, then we have T. T also you have studied, guys. Have you studied toxic shock syndrome? Yes, fatal wala. Have you studied toxic epidermal necrolysis, skin wala? Perfect. So soft is done. What is with P? P is going to be for the lung disorder, that is pneumonia, pneumatococcus. Pneumonia and pneumatococcus is going to be covered under it. In fact, let me tell you, Staph aureus is the most common cause of hospital-acquired pneumonia, also ventilator-acquired pneumonia. Lots of ventilator-acquired pneumonias that we'll be doing later on, but remember, hospital-acquired and ventilator-acquired ka most common cause, Staph aureus. Okay, soft pains. A is going to be something which is so, what can I say, it is so controversial. A is going to be causing acute infective endocarditis. Please note another most common list coming over here. The most common cause of acute infective endocarditis and the most common cause of Native valve means not prosthetic valve abhi ke liye, but native valve infective endocarditis, that is gonna be Staph aureus. Also for prosthetic valve, but that I will explain to you once I tell you all the Staphylococcus. I'll tell you a better version of all these endocarditis. But the most common cause of acute and native valve endocarditis is gonna be Staph aureus. Okay. Then pains mein I aata hai, that is going to be infective arthritis. It is associated with infective arthritis. Pains mein what is left? N and S. N is gonna be necrotizing fasciitis, which you could have anyway understood with your skin soft tissue only. Necrotizing fasciitis. And S is like a, what can I say? It's like a, a formality. Sepsis, because any bacteria can cause sepsis. So let's do a repeat. Soft pains was actually a mnemonic, which sounds lengthy, but it's so useful now when you actually start going ahead with this. So remember, skin soft tissue infections, osteomyelitis, food poisoning, TSS10, pneumonia, pneumococcal, acute endocarditis, infective arthritis, necrotizing fasciitis, and sepsis. Out of these, the picture of botryomycosis is much expected. Okay, so clinical features and virulence done. Are we ready for the diagnosis? I don't think you need to know the diagnosis. You already know the microscopy. You know this is a gram-positive cocci, and you know that this refers to the bunch of grapes. So let me show you a collage. In the entire bacteriology, you will realize I've got a lot of collages so that everything is at one place. Thoda summarize ho jaye. So I think everyone can visualize very easily how you're seeing beautiful bunch of grapes and they are purple. Purple means they are gram-positive cocci. They are gram-positive cocci and they are definitely present as a bunch of grapes. One part is done. Okay, tick mark. After that, let's come to culture. First, let's start with nutrient agar and blood agar. Let me show you one by one. If I show you nutrient agar, did I tell you it's Staphylococcus aureus? Aureus matlab golden color. So now what are you going to see? On a nutrient agar, can you see there is a goldenish color available? Do you want to go into the details of it? Okay, so why golden? What is golden? There is a pigment called staphylozanthin. They could break the word. Staphylo means it's a pigment of Staphylococcus. Xanthine means it's xanthine. Xanthine refers to beta carotene. So they will ask you which bacteria produces a beta carotene pigment at what temperature? At 22 degrees centigrade and non-diffusible. Matlab temperature to rat lo, 22 degree. What is non-diffusible? See guys, if the bacteria is here, the pigment is also here. It's not that the pigment is present everywhere in the plate. No. Wherever the bacteria is there, the pigment is there. Bacteria is here, pigment is here. Bacteria is here, pigment is here. So is it diffusing out? No. If everything would have become yellow and golden in the background, you would have said it's diffusible, but nahi ho raha. So this is a non-diffusible kind of a pigment. So remember, staphylozanthin is the name, which is a beta carotene. At 22 degree, it is non-diffusible. Each one of these is a PYQ. So this was your nutrient agar. I hope the second picture is also tick marked. Now you're left with this, the blood agar. So what is blood agar showing you? So have a look at the blood agar now. Look at this very carefully. Kaisa colony lag raha hai? Ma'am, it's looking like a dot. It's looking just like a dot. Chota sa dot. Pinhead colonies. And how much of blood is being broken? Is it like the entire area around it has become white? Nahi. I can zoom in and then I'm seeing that little tiny black area is seen. Little blood has been broken down. Means the colonies are gonna be pinhead and the hemolysis is very narrow. There is a narrow zone of hemolysis that I am seeing over here. Sorted? So what are the colonies? Pinhead colonies with a very narrow, little blood is being broken down. Beta hemolysis. This is known as beta hemolysis, right? So please note, narrow zone of hemolysis is seen. Okay, so now coming back, you have done with the nutrient agar, golden wala color, you are done with the blood agar. Now you're ready for selective media. When I say selective media, please remember, Staphylococcus aureus can grow in the presence of salt, even 7 to 10%. Selective kya hota hai, guys? Go back to your culture media chapter. What do you understand by selective? Selective means like if I am is a bias. If I am selective, I will, if as a teacher I should not be, but if I am selective, I will give some better mnemonics to few students because I want them to top, and I will not give those mnemonics to the other students. Obviously, I would never do that. But if I do that, I become selective. I'm selectively allowing some people to top and I'm not allowing the others to grow. that is a selective media. So when I give salt in this media, all of them will have some salt element in it. So if I give salt, I'm selectively allowing Staphylococcus to grow and not the others. So this selective matlab, remember the mnemonic story that ma'am is giving some mnemonics to some people, they are growing, others are not. वो होता है selective media. So mannitol salt agar, Ludlam's is also a salt agar, salt milk agar. So all salt things will come, out of which they give you a question on MSA. You know what is mannitol salt agar? Break karo. So firstly you will say, ma'am, it contains mannitol and it contains salt and of course it contains agar. So salt has made it what? Salt has made it selective. Salt has made it a selective media. Mannitol is going to tell me that is it mannitol fermentation positive or negative? मतलब difference बता रहा है. Does it become a differential media? And that's the question that you get. That mannitol salt agar is a differential plus a selective media. So you'll say, hang on, we are gonna study so many differential media, mannitol, sucrose, lactose, etc. So many fermentations we will study, but you will eventually tell me that, ma'am, now I realize that everywhere when I want to tell a difference, like mannitol ferment hua ya nahi hua, I will tell you by a color change to yellow or color change to something else. So that means that everything is going to have an indicator. That indicator will tell me that color change hua ya nahi, and the indicator over here is phenol red. So when red color will change, then I will get to know that mannitol fermented. Have a look at this picture. Firstly, mannitol salt agar. Mannitol makes it what media? Differential media. Salt makes it what media? Selective media. And guys, is it also an indicator media? So ye to trio ho gaya. This is like 3-in-1 use of mannitol salt agar. And can you see over here, because it's Staph aureus and mannitol has been fermented, what is the color that you have got? When mannitol will be fermented, you will get a yellow color. We are just starting with bacteriology. I'm telling you one trick. There are 4 fermentations that I will teach you. Okay? Everywhere I will teach you a fermentation. I will say lactose ferment hua, yellow color. Sucrose fermented, yellow color. Mannitol fermented, yellow color. So everywhere I'm going to pick up a yellow color. Where I don't pick up a yellow color, I tell you that exception. Till that time, keep mugging up yellow, yellow, yellow. So if I say mannitol was fermented and what color did you get? You got a yellow color. And the previous year question that you've got in your paper is, what is the indicator of mannitol salt agar? That is phenol red. Now your diagnosis of culture. So I can show you that collage finally. You know this is a grape-like organism, golden pigment, narrow zone of hemolysis, and mannitol salt agar pe you've got a yellow pigment. That means mannitol has been fermented. Are you ready with the biochemical tests? Remember there were 2 enzymes, Staphylococcus CC, catalase positive, coagulase positive. Jaldi se do that. Yes, it is catalase positive. How do I get to know? Let me tell you one simple trick. Whenever you will see bubbles on the slide, matlab catalase positive. That is how you solve the question in 1 second. Bubbles hai, catalase positive. Okay, but what is the concept? You know what you do? You take the culture broth and you add a little bit of H2O2. This is what I am doing. I'll take a little bit of sample from the patient. I will add H2O2. Why am I doing that? Because let's assume ke Staph aureus hai. Chalo, let's assume. Okay, Staph aureus is there. If Staph aureus is there, this means catalase is there. And if catalase is going to be there, that will break H2O2. That will break H2O2 into simple hydrogen in water, oxygen. Oxygen is going to create bubbles. So simple, if Staphylococcus aureus is there, this means catalase is there, this means bubbles banenge. If Staphylococcus aureus is not there, catalase is not there, this means bubbles nahi banenge. So simple, if bubbles are formed, it is positive. If bubbles are not formed, it is negative. But now on that note, can you tell me what are all the catalase positive organisms? So abhi, what did I tell you? I'm going to tell you a story of the bubbly cats. Why did I say bubbly cats? Because anyone who's a cat lover or a cat parent would know that cats never ever sit at one place. You can never make cats sit at one place. Cats are always going to run around the house. They're going to move around a lot. They need a lot of, they're very bubbly. Firstly, they're very bubbly. So bubbly cats tells me that cats for catalase and bubbly because when I see bubbles, it's going to be catalase. But for cats, you need a lot of space. Space being made. Guys, let's do this mnemonic of what is space being made. And let me give you that concept first, that although your mnemonics are very good, but in the exam you tend to forget A kya tha, S kya tha, P kya tha. That's a very common thing that happens to everyone in the paper. So now whenever you read your mnemonic, whenever you make a full form out of it, try to get some story into it so that it can come back to you in the exam also. So S and P, I start with all the pigment wala organisms. Right now I told you Staphylococcus aureus has a golden wala pigment. After some time I will tell you Serratia. Serratia is R na, so Serratia has a red color pigment. So repeating once again, Staph aureus has golden pigment, Serratia has red pigment, and P for Pseudomonas. Pseudomonas is gonna have a blue-green pigment. Pseudomonas has a blue-green pigment. So तीनों जो pigmented bacteria थे, I have got them. Staph aureus, Serratia, Pseudomonas, they are catalase positive. Now I'm going to get some fungal infections. A for Aspergillus, that is definitely catalase positive. C for Candida and Cryptococcus. Candida and Cryptococcus is also catalase positive. Now E के साथ दो चीजें. There is going to be E. coli and Enterobacter. Basically, there's an entire family called Enterobacteriaceae family. So that Enterobacteriaceae family is going to come up over here. That is space. ठीक है, तो space का तो I have understood. SP for pigments, AC for all fungal, E for E. coli Enterobacteriaceae family. Okay, now let's talk about space being made. When I'm talking about being made, these are the things that I'm gonna pick up. So B say we will pick up the very deadly Bacillus anthracis, and NG is not only gonna be Neisseria gonorrhoeae but also the other Neisseria, that is Neisseria meningitidis. तो दोनों जो Neisseria है, they are going to come under it, right? So Neisseria gonorrhoeae, Neisseria meningitidis. Space being made almost towards the end. So M is for the very famous Mycobacterium tuberculosis. And M, I think you're forgetting. If you've kept Staphylococcus, how can you forget the cousin brother of his family, and that is Micrococcus? So that's the last one that you have to know. I know it's a lengthy mnemonic, but it solves so many problems for the rest of bacteriology. So pehle pigment wale, then fungus wale, then E. coli Enterobacteriaceae, Bacillus anthracis, Neisseria gonorrhoeae meningitidis, Mycobacterium TB, and Micrococcus. All of these are bubbly cats. They are catalase positive. Okay, so catalase done. What are we left with? We are left with the next enzyme, and that is the coagulase test. अब पहले तो रट लो, then I'll tell you the logic. ठीक है? Coagulase test can either be done in a test tube or it can be done in a slide. So I call it slide coagulase, tube coagulase. There are other names for it. Tube coagulase is known as free coagulase, slide is known as bound, and students go in the exam and forget this. So बहुत simple. Tube has how many alphabets? 4. And free has how many alphabets? 4. Slide has how many alphabets? 5. Bound has how many alphabets? 5. So 4 4 एक साथ, tube and free are the same, 5 5 एक साथ, slide and bound are the same. रट्टा हो गया? Now these are gonna be positive in almost same. दोनों Staph aureus show you tube and slide, Staph intermedius shows you tube and slide, Staph hicus shows you tube and slide. So you'll say difference का है ma'am? Look at this. Staph lugdunensis only shows you bound, it doesn't show you free. And Staph schliefferi only shows you free. हो गया confusion? चलो let's sort. Staph schliefferi has the word schlieffree. From today onwards, call it Staph schlieffree. So you will know that, okay, schlieffree में कौन सा coagulase? Free coagulase. And lugdunensis over here is gonna sound like bound. So this is lugdunensis, bound. That is gonna have bound coagulase. Otherwise, Staph intermedius, hicus and aureus, they are gonna actually have both. But lugdunensis has bound and schliefferi has free. Ratta ho gaya? Should I tell you the logic behind it? Let's start with the first one. Look at this. I've told you a tube coagulase. You'll say, ma'am, so test tube to is mandatory. I will get test tube mein. And this is what? Tube 1, 2, 3, 4. So this is free 1, 2, 3, 4. Ye coagulase already hai. So let's assume that I have taken a Staphylococcus ka culture broth. Will that have coagulase? Of course, Staphylococcus hai. It will have free coagulase. What will I do? I will add a little bit of my human plasma into it. Human या कोई animal का plasma, because all our plasmas have something called CRF, coagulase reacting factor. Concept बन रहा है that if Staphylococcus has coagulase and I add coagulase reacting factor, तो combination में clump बनेगा. Coagulase reacting factor from my plasma and coagulase from the Staphylococcus aureus is gonna make a clump of a CRF coagulase complex. So CRF coagulase ka complex banega. As soon as this gets complex, you know what happens? you had added plasma. Plasma has fibrinogen, clot ban jayega. This is going to activate the fibrinogen and there will be clotting of the plasma. So now if you look at these two test tubes a little carefully, you can very well see that in this there is a coagulum or a clot that has formed. This means this is a positive tube coagulase test. So the only question that you get, tube mein 4 alphabets, free mein 4 alphabets, because the coagulase is totally lying free in the Staphylococcus. Second, they will ask you that in which one do you use the coagulase reacting factor? Coagulase reacting factor is used over here, and how do you assess it? By the clotting of the plasma. So ye tube wala tha. What is gonna be the slide wala? Firstly, 1, 2, 3, 4, 5. Slide wala is 1, 2, 3, 4, 5. Bound coagulase means it is bound to the cell wall. Whenever something is bound, like for example, if this is staph, this coagulase is bound. This pencil of mine is coagulase. ये bound है. Bound मतलब clump हो गया. Bound मतलब clump हो गया. So this is also known as the clumping factor. And I can see that to this, as soon as I will add a little bit of fibrinogen, it is going to result in complete clumping. As soon as I will add fibrinogen, jaldi clumping ho jayega. But yeah, there are differences in the timing of a tube and slide, but all those details I'm sure you don't want to go into because it's unnecessary, not required. But please note that they will ultimately ask you questions, which is tube, which is slide, which is free, which is bound, wo ho jayega. Second question, they will ask you, where do you use coagulase reacting factor? So coagulase reacting factor is going to be used in the free wala, whereas clumping, how I have learnt is clump matlab clump ho gaya, bound ho gaya. So clumping factor is going to be used in the bound or the slide coagulase. That wraps up. Let me take a test. Bachchon wala test. Is it tube coagulase or slide coagulase? Ma'am, common sense. Kya question hai? This is tube coagulase. I can see a tube, and this one is positive because I can see the clot also that has happened. So tell me, tube matlab free or bound? 4 alphabets. Ma'am, this is free. Tell me, this is clumping factor or coagulase reacting factor? So bound hota to clump hota. This is coagulase reacting factor that is going to be needed. Sorted for everyone. Now let's go on to the final diagnosis. There are two types of typing that you do. Abhi tak to culture and biochemical is done. What is the typing? Please note there is something called phage typing and genotypic typing. Phage typing matlab I am going to use bacteriophage. So you don't have to know the details of phage typing. You just have to know that I am trying to find out, this is staph and this is bacteriophage. How can this bacteriophage kill it? Or how is the susceptibility? On those bases, I realized that phage 80 and 81A have been associated with outbreaks. The staphs which result in outbreaks, they are susceptible to phage 80-81. But jo sabse important question hai, phage 3 is seen in association with MRSA. Ye thoda ratta hai that you will have to do. I have not made any mnemonic because how many will you learn? So please remember, phage 3 is associated with methicillin-resistant Staph aureus. On the other hand, there are some genotypic methods also. There is a pulse field gel electrophoresis that can be used. There are many other methods which they don't ask you. But yes, pulse field gel electrophoresis is a genotypic method for typing the Staph aureus. Diagnosis ho gaya. If I have to give you a very quick recap, you first did the microscopy, then nutrient agar golden color, blood agar you did narrow zone of hemolysis, mannitol salt or anything with salt becomes a selective media. Then you will pick up that bubbly wala test that is going to be catalase positive. Then you pick up a test in a tube that is going to be tube or free coagulase. Then you pick up a test in the slide and that is going to be slide or bound coagulase. Then you finally do a phage typing in which phage 3 is for MRSA or a genotype phaging which is going to be a genotype typing which is a gel electrophoresis. So you are now ready for what? You are ready for the treatment aspect. And you know beta-lactams or penicillin are going to be the drug of choice. So beta-lactams or penicillin is what is going to be used over here, right? Okay. Mechanism of action. Mechanism of action is, I've drawn this one tiny little thing. I hope everyone can see, hai na? So does this remind you of something? NAM, NAG, kuch yaad aa raha hai? Yes. What is NAM, NAG? Ma'am, this must be the cell wall. And you had also told us, ma'am, in general that NAM are connected. NAM are always connected. Yes, these are known as the penicillin binding proteins. There are something known as PBP. You really don't need to know the full form, but yeah, you can because it's kind of obvious. Penicillin binding protein 1, 2, 3, 4 are present. These are also known as transpeptidases. These are also known as transpeptidases. These are the places where the penicillin is going to go and act. So what is the penicillin going to do? Penicillin will bind obviously to penicillin binding protein or transpeptidases, and penicillin will break this wall. So simple, bacteria gaya, cell wall gaya, so the bacteria is also gone. But do you think the bacteria is that simple, that innocent? Now no bacteria is that innocent. Now we have a lot of resistance. So what does the bacteria do? Bacteria says, hang on. Ab mera time hai. So now the bacteria is going to start with the story of resistance. How is resistance maintained? So bacteria says, hang on, what is coming to kill me? Beta-lactam is coming to kill me. I will make beta-lactamase. Before beta-lactam kills me, I will kill beta-lactamase. So the resistance is going to be by beta-lactamase production. The bacteria starts producing beta-lactamase. You will ask me, aise kaise ma'am? Without genes, how can it produce? Yes, this is going to be regulated by BLA, beta-lactamase. So matlab this is regulated by BLA gene, and this is going to be regulated by PLA, that is plasmids. Okay? So remember that beta-lactamase production, that is BLA, is going to be regulated by the BLA, that is BLA gene, and that is transferred by PLA, that is plasmids. So transfer kaise hoga? If there has to be a transfer of resistance, there are two methods known. I'm going to make a box because this is quite controversial. One of the methods is going to be transduction, and the other method is going to be conjugation. So if they ask you, theek hai, dono to theek hai, but which is the commoner one? So for Staph aureus, I know, as soon as I say plasmids, I know what's coming to your mind. That in bacterial genetics, you told me, ma'am, plasmid matlab conjugation hoga. For all other bacteria, conjugation is more important. But for Staph aureus, the one that you study in maximum detail, trust me, don't get scared of bacteriology by looking at the length of Staph aureus wala chapter. Staph aureus is lengthy. After that, things become chota. Okay? So please remember, for everywhere, conjugation is more common, but transduction is more common in Staph aureus. That is what I want all of you to make a table around, because this will come in the paper. Okay? So now you've understood. You tried to kill with penicillin, but bacteria said, I'm smarter. I will make beta-lactamase by BLA gene, by plasmid, and I will kill the beta-lactamase. So now if you are resistant to penicillin, what are you going to give to the patient? You're going to give methicillin. Okay? But bacteria is again smarter. Bacteria says, I will become resistant to methicillin also. What is that known as? Methicillin resistant Staph aureus. M for M. How did it become resistant to methicillin? That is by the MEK-A gene, and this is going to be chromosomally mediated. This is chromosomally mediated. That is why I told you that pichla wala was blah blah blah, because you know that is plasmid mediated. This becomes chromosomally mediated. And hota kya hai? Did I tell you that all of these have to bind to a penicillin binding site? Penicillin aayega, it will bind here. Methicillin aayega, it will bind here. So the penicillin binding site 2 is altered to penicillin binding site 2A. Now this initially could bind to methicillin, but PB2A has a lower affinity. It will not bind to methicillin. It is not going to bind to methicillin. So that is what I've said. PBP is going to be altered to PBP2A, which is going to have a lower affinity. Okay. Now you say that, acha, ye bhi resistance ho gaya. Ma'am, how do you find out? How do you find out in a patient, in a sample, that methicillin has come out to be resistant? I'm going to tell you a series of things. But the first one that I'm telling you, no one does it, but MCQ ke liye best hai. How do you do the diagnosis of methicillin resistant Staph aureus? Best method is do a dilution. Remember when I was telling you antibiotic sensitivity, I told you that there is dilution broths created. So which antibiotic do you test? You test against cefoxitin and oxacillin. Now first question, big confusion hua. Ma'am, you were wanting to find out methicillin resistance, but in the lab, the test you are doing is for cefoxitin and oxacillin. Aisa kyun? Why are you not testing in the lab for methicillin only? Because methicillin as a drug is very unstable. You cannot test it. So for testing of MRSA, we actually use cefoxitin and oxacillin, and they say that on dilution broth, if you need, what have I written? MIC. Guys, put your brain. MIC kya tha? General microbiology. Quickly, minimum inhibitory concentration. Matlab, what is the minimum amount of antibiotic that I need to kill the bacteria? Like, for example, what is the least amount of information that I have to give to you for you to clear the exam? If I don't give you that much information, or I know how much of information to give you, less of that also will not make you pass. Excess of that also will not make you pass. I know exactly how much information is needed. So exactly what is the minimum amount of antibiotic that will kill the bacteria? That is minimum inhibitory concentration. So they say if the MIC of cefoxitin is more than 8, matlab 8 se bhi zyada if it needs, means it is resistant. And if it needs more than 4 micrograms of oxacillin, then also it is resistant. Matlab beyond this, even if it's needing even higher quantities of these antibiotics, matlab it's definitely resistant. So the MIC of cefoxitin is 8 and the MIC of oxacillin is going to be 4 microgram. This is the best method on paper. Karta koi nahi hai. What is done? Rapid methods are done. They do disc diffusion tests. Again, they use the same thing. They use cefoxitin and oxacillin, and if in case they ask you which is the better one, cefoxitin is always better than oxacillin, just in case the question comes. Other than that, they can do even rapid tests like latex agglutination, ELISA, PCR, ye sab for knowing the genetic problem. These are all rapid, more faster, easier to perform rather than doing dilution broth. But yeah, exam mein question hai, so dilution broth is going to be the best. But before I proceed, let me tell you, for the diagnosis of MRSA is very, very important in all of us, in healthcare professionals, because you know why? What is the most common mode of how methicillin-resistant Staph aureus spreads? It's by our hands. It's the hands of the healthcare workers. It is by our hands. That is why they say that the precaution is that you have to, before and after every patient, you have to do hand washing. Because you've dealt with the patient, you might transfer that MRSA onto someone else. So the rigorous hand washing is very important because we are the people who have the maximum chance of spreading this MRSA. But just in case you do a hospital study and you say, ma'am, I found that 10 nurses and 20 doctors were having MRSA. Where? So remember that the most common site that you have is usually nasal carriage. So if you find the nasal carriage or the people who are carrying Staph MRSA in their nose, then you have a nasal ointment known as mupirocin. You have to give them for 5 days, both the nostrils they have to put. So there is a nasal ointment if they are nasal carriers. But for others, you can do some washes of chlorhexidine. For other mean other sites, not nasal carriers, but if they are doing MRSA carriage in some other site of the body, chlorhexidine washes. Is that okay? So you've done everything for your healthcare professionals also. MRSA, we are the people who spread it the most, so we have to wash our hands. If we come across any nasal carriers, we have to give them an ointment of mupirocin. So this is done. But now, when you were penicillin resistant, you give methicillin. Now you've become methicillin resistant also. So what are the drugs that are available? There is vancomycin. There is daptomycin. These are the drugs that are available in case of methicillin resistance. But now let me tell you the further part. Now we are also vancomycin resistant. Resistance so chalta rahega na, because with so many antibiotics being used without any proper evidence, resistance is gonna come up, and there is gonna be a time that we are going to use the high-end antibiotics because bacteria will stop responding to all of these. So now even vancomycin resistance is known. That is known as VISA and VRSA. VISA and VRSA matlab SA to pata hai, Staph aureus. So VISA basically refers to vancomycin intermediate Staph aureus or intermediate Staph aureus, whereas VRSA refers to vancomycin resistant Staph aureus. There is a difference between VISA as well as VRSA. Why? VISA matlab it is on its way to resistance. Abhi kind of it has started becoming resistant, but VRSA means definitely resistant. How do I get to know? I will do the MIC. If the MIC if you need 4 to 8 microgram, minimum inhibitory concentration of vancomycin is between 4 to 8 microgram, it is VISA. But if you need even above 16 microgram, even at 16 microgram and above, the bacteria will be killed. Then to matlab it has become such a high dose is needed. Then it has definitely become resistant. So even if you don't learn the previous wala MICs, this you have to learn because this is a definition that has been put up by CDC. Remember, VISA ke liye it has to be between 4 to 8, and VRSA ke liye the MIC is almost double. It has become above 16. So what is the difference? How does the bacteria first become VISA and then it becomes VRSA? So if the vancomycin said that, okay, I have to go and bind to the cell wall. Vancomycin diya, vancomycin say I will go and bind to the bacteria cell wall. Bacteria cell wall say मैंने तो अपना structure बदल लिया. I have changed my structure. I have altered my cell wall and my structure. You can't bind to me, so I am escaping. That is one way. Now that is when the organism will become VISA, because bacteria is kind of started ignoring the drugs, kind of changed its structure and kind of ignored the drug. However, when does it become VRSA, vancomycin resistant? When, please note and go all the way back to your chapter. Do you remember in general microbiology, I taught you that all cell walls are made up of D-ALA, D-ALA. You know what the bacteria says? I will change my name only. If I change my name, you will never be able to reach me. And that says, the bacteria says, I am now gonna change from D-ala to D-lactate or D-ala D-serine. I will now change my name only. How will you identify me? तो कैसे change करा? By the VAN-A gene, vancomycin gene ना, by the VAN-A gene. So देखो दो things है. Say for example, vancomycin is that very irritating relative of yours. ठीक है? That relative is coming to your house. You don't want to meet that relative because he will always ask you how many more attempts will you give, when will you give FMG, NEET, PG, when will you get married. All those irritating questions are gonna come. So you have two options. Your mother says, देखो, you will have to meet them, but you can say hello and then go inside your room. This means you are becoming visa. Means little bit you are gonna meet them, little bit you have altered your own habit. You have said hello and you have gone inside your room. Second is you have become immune and you are saying, to hell with all my relatives. I hope none of you show it to your relatives. But yeah, to yours, you're like, I don't want to meet them only. I'm not going to face them only. You have become like totally resistant. What did you say? You will say, tell them I don't live here. Tell them I'm in the hostel. Tell them I'm at a friend's place. You have changed your address. You have changed your name only. Now your relatives will never be able to catch you. This means now you have become VRSA. ठीक है? तो visa is where you're kind of trying to escape from them, and VRSA is when you've totally escaped from them. How? By changing your address. So the bacteria changes D-ala to D-ala D-lactate. अब हमेशा याद रहेगा. Yes, that is sorted. Staph aureus is sorted. But now I want to talk about the last part of Staphylococcus. Please note, whatever I was teaching you up till now was COPS. COPS means I was teaching you coagulase positive staph. I was teaching you coagulase positive staph, because now I'm going to teach you cons. Cons obviously becomes coagulase negative staph. And there are two very important ones that are gonna come under it. That is gonna be what? It is staph epidermidis and saprophyticus. What did I say? Epidermidis and saprophyticus. There are many more. For that matter, even lugdunensis sometimes they say comes under it. But अभी हमने पढ़ा कि lugdunensis में कौन सा coagulase हो सकता है, bound हो सकता है ना? So there are a lot of variations. You will realize main पढ़ना है. That is the first rule of microbiology. ठीक है? Staph epidermidis and staph saprophyticus. Which is more common? Staph epidermidis is the more common one that you find. And what does it cause? It usually causes prosthetic heart valve endocarditis. You will study that in heart. In pathology also I've taught you. Staph epidermidis causes prosthetic heart valve. Saprophyticus on the other hand is usually famous for causing UTIs. It is a UTI causing organism. Now remember that epidermidis is novobiosyn sensitive and saprophyticus, again I've always told you, जिसमें ये F वाला sound आता है ना, वो tough हो जाता है. So that is gonna be tough, resistant. So remember when I told you bacitracin शुरू में, bacitracin between micrococcus and staphylococcus, I told you staphylococcus is tough. So bacitracin resistant and micrococcus was sensitive. यहाँ पे novobiosyn is resistant in saprophyticus because that has F in it, tough. So novobiosyn is sensitive in epidermidis. But now I will tell you that which of them can show you a biofilm production, that is staph epidermidis. And listen to me very carefully. What is biofilm? Biofilm is nothing but made up of polysaccharide matrix. It is going to be made up of a polysaccharide matrix, and it basically protects the bacteria against antibacterial drugs or antibiotics. So any bacteria that has it always doesn't make a biofilm. Sometimes, for example, you will say that, ma'am, I have seen that there is a growth of staph epidermidis in someone's IV cannula. And that is why, the antibiotics are not reaching that person, or there is a growth that is happening, staph epidermidis. Epidermidis makes a shield. You will try to kill it with an antibiotic, no effect, because it has made a shield. Sometimes it can make a shield, sometimes, not always. Biofilm can be made by staph epidermidis. And on that note, we have sorted all of our staphylococcus, but we are all ready for some questions. Which of the following is incorrectly matched below between staphylococcus and micrococcus? याद है, I gave you one mnemonic that micrococcus में सब कुछ OO होगा. Is it oxidase positive? Correct statement. Okay, is staphylococcus then oxidase negative? ये भी correct statement. Now, then I told you that staphylococcus में सब F होगा. Is it going to show you a fermentative pattern? Correct statement. Is micrococcus going to show you a fermentative pattern? No, micrococcus में सब कुछ O था. What glucose pattern will it show you? It will show you an oxidative pattern. So this was the false and this was the answer, because they asked you the incorrect statement over here. Coming to the next. Which of the following organism does not show the test below? Does not. ये कौन सा test है? Bubbly, ma'am. This is the bubbly cat test. Bubbly cat test means it is catalase, and for catalase we had a mnemonic. For catalase we said there is space being made. Space being made में Staphylococcus was there. M का Mycobacterium was there. P से Pseudomonas was also there. What was not there? Streptococcus. So which of them does not show you catalase positivity is your next chapter, that is Streptococcus. Moving on to the next. Which of the following organisms does not show the test below? कौन सा test show कर रहा है? Tube coagulase. Tube का दूसरा नाम 4 alphabets. तो free coagulase test. Okay, free. तो free में पहले तो tell me Staph aureus मैम ये तो tube और slide shows both. Then Hicus intermediate they show both. Then I realize Schlie-Ferry has free, so this also definitely shows free. ये भी होगा. But Staph lugdunensis is usually either it's not showing coagulase, and if it's gonna show coagulase, it's gonna be bound coagulase, right? Bound, because lugdunensis I told you na bound की तरह. Bound मतलब 5 alphabets. The slide coagulase test. Coming on to the next one. Indicator for mannitol salt agar. I told you this is going to come across as a previous year question. Indicator for mannitol salt agar is phenol red. Simple one-liner. And coming to the last question now. What is mannitol salt agar? Firstly, is it a selective media? Yes, salt makes it selective. Is it also a differential media? Yes, because mannitol makes it fermentation hai, nahi hai. It is differential. So what is it? The answer becomes both. It is both selective and differential media, and that is definitely the last part of Staphylococcus.